let's get started. So yeah, thank you everyone for coming. Before I introduce our super exciting seminar speaker, let's just take a moment to acknowledge that we're on coastal Miwok land and the historical and ongoing colonial violence of Indigenous people in many places. Yeah, so with that, now I'll introduce Dr. Alicia Abadia Cardoso is a professor at Universidad Autónoma de Baja California in Ensenada. She did her master's there in Ensenada and her PhD at UC Santa Cruz in collaboration with NOAA Southwest Fisheries Science Center in Santa Cruz, where she did her postdoc as well. Her research focuses on applying molecular genetic methods to answer ecological, evolutionary, conservation, and management questions on aquatic species, from marine mammals to commercially important species. And specific goals are to promote the conservation of biodiversity by evaluating the population's genetic variability, inbreeding levels, taxonomic uncertainties, and the effect of fishing, pressure, and habitat fragmentation. And today she will be, yeah, I guess I was going to read the talk title, but there it is. So thank you so much for coming.
Thank you. I think I turned this on. Can you hear me okay? All right, cool. Well, hi everyone. Thanks to Shreya for the invite to this seminar series. And thank you for being here today. So I'll talk about the use and application of genetic tagging. And particularly, I'm going to talk about parentage-based tagging, which is a genetic tool that we use for the study of species. Specifically, I'm going to talk about aquatic species. So in this talk, I'll start briefly describing genetic tagging, and then what do I mean with parentage-based tagging. I'll tell you about a case study where parentage-based tagging was used for a pedigree reconstruction in steelhead. And then I'll talk about a project that is in progress and that we are planning on using this same method as another application for conservation of a threatened species, which is the black abalone. So we know that a first step in conservation and management of any population is first to understand its biological patterns, life history traits such as survival, reproductive strategy, reproductive success, etc., are especially critical because they shape the direction of evolution of the population and its responses to environmental changes. But these life history traits are extremely hard to measure in species that have complex and evasive life cycles and where direct observation or direct tagging methods have become very complicated. And they can be very invasive too. You have to trap the individual, sometimes for long periods of time. And in many cases, it is necessary to recover the tag, and that implies to immobilize the individual again. Another problem is when the tags are lost. For example, species that live in the intertidal that are exposed to high energy tend to lose the tags very easily. And so this is why genetic methods have become a fundamental tool for the study of these species. So these molecular or genetic tools allow us to address multiple issues, such, just to mention a few, sex determination, paternity and kinship, migration and movements, food preferences, forensic applications, population structure, and connectivity between stocks, bottlenecks, phylopatry, among many others. They are very, very powerful. So these genetic tools starts with sampling, and sampling to obtain the genetic data begins by obtaining biological material, such as biopsies, small pieces of pin or tissue, skin, blood, hair, vibrisses, feces or mucus, and we can even collect water that contains cells to do this genetic analysis. So this type of sampling has multiple advantages, including that it applies to any species, so it is universal. Also, any type of biological sample that contains DNA can be used. The semi-invasive methods that are used to take the samples or tissues allow us to do it without damaging or stressing the individuals too much. And furthermore, it is non-lethal. That is not necessary to kill the individual to obtain the sample when we're talking about multicellular organisms, of course. Also, once the genetic data is obtained, it is not lost like a tag that can fall from the individual or the label that fades over time. And finally, this allows the identification of the individual without ambiguity. And with these biological samples, what we do is that we extract the DNA to obtain molecular markers. So what are the molecular markers? They are fragments or pieces of DNA that we extract from the biological sample and that allow us to evaluate the variation in the nucleotide sequence of that DNA fragment between one individual and another. And there are many molecular markers that have been developed since the 80s or earlier. And in this list, I'm just mentioning just a few. And among them, the most used in recent decades are the microsatellites and sequencing. And more recently, with the development of throughput sequencing methods, the single nucleotide polymorphisms, or SNPs, have become very important and then derived from those are the microhaplotypes. So the work that I'm going to present today focuses on these SNPs, and I'm going to tell you some of the applications. So what is a SNP? A SNP is a type of genetic variation between two or more DNA sequences when a single nucleotide changes, and it is normally biallelic. That means that we have two options. For example, in this example, I have two individuals. Individual 1 that has its DNA sequence from 1 to 10 nucleotides, and individual 2 that has also its sequence. And as you can see, in position 5, individual 1 has a T, and individual 2 has a C, and that's a SNP. And you can analyze multiple SNPs along the genomes that you are working with. But why are they so great? Well, they have several advantages that make them very useful. Just to name some, they are widely distributed. It's the most common type of variability in the genome. They occur in coding and non-coding regions, and this is important depending on the goal that you want to get to. They have low mutation rates compared to other markers like microsatellites, and we have a good understanding of the mutation models. Also, data obtained from SNPs are easily portable and combinable between labs, and the new technologies for genotyping SNPs now allow us the examination of a large number of SNPs in a large number of individuals and in short time periods and at relatively low cost. So now I'm going to talk to you about this case study in which we evaluate patterns and heritability of life history traits of steelhead trout using parentage-based tagging, which is an application of genetic tagging. This study began in 2007, and it is a work that has involved more than 15 years of sampling. So it has been a very important effort in which many people have collaborated, mainly from the National Marine Fisheries Service from NOAA in Santa Cruz, the Fish and Wildlife Service of California, and the University of California. I have been involved in this study from the beginning in 2007 as a PhD student, and now I continue to collaborate in this effort with another PhD student, Ant Wilkie, from the University of California in Santa Cruz. Among salmonids, Oncorhynchus mykiss has perhaps the most complex life history. Two major ecotypes can be distinguished: the non-anadromous resident type called rainbow trout that can spend the whole cycle in fresh water. And then we have the anadromous type called steelhead. After hatching, steelhead spends 1 to 7 years in fresh water and 1 or more years in the ocean before returning, usually to their natal stream, to spawn. On top of this variation in timing in ocean entry and reproductive maturity, steelhead may also be heteroparous. That means that they can spawn in more than 1 year. And this complexity makes them very difficult to understand their biology and obviously manage their populations. So such management or assessment has become increasingly important since many steelhead populations have declined, and many are now protected. The most important cause for this decline include habitat loss and degradation, recreational harvest, and artificial propagation, followed by supplementation like hatchery programs. We know that artificial propagation is a common method for addressing population declines. However, artificially propagated populations may face a range of problems. For example, artificial selection, inbreeding depression, increased disease susceptibility. And this can have an effect on the natural populations like maladaptation and reduction in fitness due to introgression by stocked individuals. So, to minimize these effects, it is necessary to reduce artificial selection, and to do that, it is necessary to understand in detail the population life history traits. So the first goal of this study was to provide a level of detail on the biological population patterns, such as heteroparity rate, age structure of the returning adults, variance in reproductive success and sexual maturity, variance in family size, and we wanted to look if there was a correlation between families and spawning time. And all this was necessary for the construction of an effective management program for the hatchery mitigation plan. So also, this study documents the utility of SNPs for parentage-based tagging. But how to do this in a complex organism such as Oncorhynchus mycis? Well, fortunately, the combination of recent improvements in molecular markers and statistical analysis have provided a more accurate and cost-effective alternative to direct mark recapture studies using the genetic analysis. So this genetic tagging consists in getting a thin clip from the adults in the population and obtain their genotypes. This can be the adults in a stream or a river, or in the case of this study, from individuals that return to the hatchery to spawn after their ocean migration. So these genotypes that are a combination of molecular markers, like the SNPs, are represented here in the colored squares. And each combination of genotypes is unique for each individual, so it's like a fingerprint. So these first genotypes allow us to get some information about the population, such as genetic diversity, effective population size, kinship, and even population structure if you are looking at different locations. But these were not the goals of this particular study. Here, the adult sample migrates to the ocean, and they can come back to the hatchery the following year. When they come back, they are sampled and genotyped again, and that way we can obtain the heteroparity rate. On the other hand, the offspring from these adults are also released at the age of 1, and they migrate to the ocean, and they can spend a few years there. Then they come back to the hatchery as adults, where we obtained their genotypes, and with this information, we can reconstruct pedigrees with the parents that were previously sampled. Because we already have the genotypes of the parents, we can infer the genotypes of all the offspring. So that way, we know when were they born and the age at which they came back, so the age of maturity, and we can also estimate the parents' reproductive success, family sizes, among other things. So this study was conducted in the Russian River, you are all familiar with. The Russian River is located in north central California, and this basin maintains populations of coho, chinook, and steelhead, and these populations have declined dramatically in the past few decades. And the decline has been attributed to anthropogenic activities, particularly 2 dams that were constructed on the Russian River, one in 1959 and the other in 1982, had an impact on these salmonid populations, species. Therefore, hatchery programs were established to mitigate the decline of the fish populations due to dams. These were the Coyote Valley Fish Facility and the Warm Springs Hatchery. Also, I wanted to tell you that both the wild steelhead population and the fish produced at the hatchery are part of the Central California Coast Distinct Population Segment and are listed as threatened. So from 2007 to 2020, roughly 11,000 fin clips were collected from all the adult winter steelhead that were artificially spawned at Warm Springs Hatchery. And from 2009 to 2020, about 6,500 were collected at Coyote Valley Fish Facility. So based on the steelhead migration patterns that I told you about, we know that fish that spawned in 2007 can be parents of fish returning in 2009, 10, and 11. And fish spawned in 2008 are potential parents of fish returning in 2010, 11, and 12. And fish that spawned in 2009 can be parents of fish that return in 2010, 12, and 13, and so on until 2020. So all the individuals were genotyped with 95 SNPs. And this can be very small for the people that do genomics and analyze thousands and thousands of SNPs. But in the study that we did in 2013, we demonstrated that only 95 SNPs were enough to do a pedigree reconstruction and with that high confidence in this population. So since the steelhead have the potential to spawn on multiple locations, so they are Iteroparas, spawn adults that are released into the river and may return and participate in additional spawning events in subsequent years. So to account for these situations, the R-package hatchery pedigree was created, and this identifies fish that spawn multiple times through a matching samples analysis. And with this, we identify the Iteroparas fish. Then for the pedigree reconstruction, we consider adult fish spawned in 2007 to 2018 as the potential parents of fish returning from 2009 to 2020. And we assigned parentage, so we reconstructed the trios, the parent-offspring trios, using the R-package snippet, which implements a likelihood-based method for large-scale parentage analysis using these 95 SNPs. And by using the inferred parent-offspring trios, we determined the age of the returning adults. We also calculated the distribution of family sizes. The reproductive success was estimated by counting the number of offspring that returned to the hatchery per parent. We also estimated the heritability of date of spawning during the season, and it was calculated using a parent-offspring regression. And we also calculated the heritability of the age at reproductive maturity using a Crittenton-Falconer test. We also evaluated the impact of inbreeding on fitness, and we did this counting the number of offspring that returned to the hatchery to spawn per parent pair that had some level of relatedness. So for the matching samples analysis, we first calculated the probability of identity, which is the probability that two individuals would have identical or nearly identical genotypes at 95 SNP loci, given the allele frequencies of the population. And this probability was extremely small. The probability of identity of full siblings was 10 to the minus 10 and 10 to the minus 23 for unrelated individuals. So samples with identical or nearly identical genotypes were considered to be from the same individual. And with that, we found 68 Iteroparas fish. All of them returned only twice and in consecutive years. And this corresponds to 0.4% out of the total number of individuals spawned from 2007 to 2020. And this result is slightly lower than other studies that report around 1%. We also observed that the proportion of Iteroparas males and females was statistically different, being more common in males. And this is different than what we see in other studies, where Iteroparas females are more common than males. And our result could be explained in part because we have a greater number of males in our samples. And this is because of the artificial spawning that is happening at the hatcheries. And on the other hand, it is possible also that artificial spawning in the hatcheries eliminates competition between males, and that increases the survival rate and also the frequency of Iteroparity. So the final number of offspring assigned to parents or reconstructed trios was 13,474. So the results are shown in more detail in these two tables. In the first table, we have Warm Springs Hatchery, and in the second table, we have Coyote Valley Fish Facility. And the first column is the spawn year. Then we have the total number of fish sampled, and then the total number of fish that were assigned to parents. And the last column is the percentage of assignments. So the total number of fish from Warm Springs Hatchery in 2009, I forgot that 2009 was 217 fish assigned, that's 34.3%. And this seems small, but remember that fish spawned in 2009 can be offspring of fish spawned in 2006 and 2005, and we didn't have those samples. Also, if you look at Coyote Valley in 2009, in 2011, sorry, we have 249 fish assigned to parents, and that's 35.7%. That also seems very small, but it's the same thing. Fish that were spawned in 2008 or 7 could be parents of these fish. But if you look at the numbers in 2010 in Warm Springs Hatchery and in 2012 in Coyote Valley Fish Facility, you see that we reach about 80% of assignments, which is pretty good, and every year it increased. So for the age at maturity, in this plot, I'm presenting the proportion of females and males that return to spawn. That means that they reach reproductive maturity at the ages 2, 3, and 4. And we did not find any 5-year-olds. And we can observe a great proportion of females that reach maturity at 3-year-old. And while males, although most of them return at age 3, we have a large proportion that return at the age of 2. This is very interesting because it shows a trade-off between fecundity and survival in steelhead ocean migration. Fecundity, we know that fecundity in females increases with body size, which generally increases with age. And moreover, if we look at this other graph, where we have the total length from all fish that were sampled from 2013 to 2020, is when we started measuring them, and the relative frequency, we see that 2-year-olds are smaller than 3 and 4-year-olds. Also, we know that access to marine resources allows a faster growth, but spending more time at sea also increases the risk of predation and obviously reduces survival. So these results on this sex trade-off, dependent trade-off, were males in which fecundity does not depend on body size, benefit from spawning at younger ages, while females benefit from spawning at older ages. Now for the age distribution of the returning adults, in this plot I'm presenting the timing of adult returns within the spawn season by age. On the top plot we have the females and on the bottom the males. And each column corresponds to a 5-day interval in which fish spawn within the season. So from day 1 to day 120, which is the 3 months that the spawning season lasts. And the colors is 2-year-olds is brown, 3 is blue, and 4 is orange. And we see that 2-year-olds spawned later in the season, here, than 3 and 4-year-olds. And this pattern is the same for females and males. And this could be because 2-year-olds have not reached maturity at the beginning of the season, and so they do it until the end of the season. And well, this is just a speculation, but it's a probable, possible explanation. So now about the distribution of family sizes, it was highly variable. In this plot, the different colors represent the parents. So we have the female parents, male parents, and the parent pairs. And on the x-axis, we have the number of offspring, and we see that around 55% of the families were one offspring families. And the average number of offspring per parent pair was 2, ranging from 1 to 39. On the other hand, we can also see differences in reproductive success. between females and males. Females showed a higher reproductive success. The average number of offspring per female was 4.2 and 2.5 per male. Now, we found a strong positive correlation between the day of spawn of the parents and offspring across all years. This is the graphic representation of the regression. On the x-axis, we have the parents' spawn date, and on the y-axis, we have the offspring spawn date. And there is some variance in the data points. However, the correlations were strong and were not different between sexes or between 2-year-olds and 3-year-olds. And we found a very high heritability in the strict sense. 0.73% is really high. And this is interesting because, as I mentioned before, we observed that 2-year-olds spawn later in the season than 3-year-olds. And this could be related to age at maturity. In 2013, we did not have enough data to test this, but with the data the following years, we did it. And this is what we found. Here on the x-axis, we have the age at sexual maturity of the offspring, meaning the age at which the fish returned to spawn. And on the y-axis, we have the proportion that corresponds to the mid-parent age that we have in the different colors. So as we can see, the proportion of age to offspring from age to parents was higher than the proportion of age to offspring from older parents. While there is a larger proportion of offspring that return at age 3, with parents that return at age 3 or older. So, with this data, we estimated the heritability in the strict sense of the age of maturity using a subset of parents and offspring trios for which we knew the age of all of them. And once again, we got very high heritability values, 0.41. And there is a strong genetic component then that determines the reproductive maturity. And to give you an idea on how high this value is, the mean H2 values for various life history characters in the genus Oncorhynchus is 0.24, and in other salmonids from 0.16 to 0.27. So these are very high heritability values. So finally, since heritability of a spawn date could cause an increase in the incidence of related individuals spawning on the same day, we examined the impact of inbreeding on survival, on the offspring survival. So we found that inbreeding does have a potential impact on fitness based on offspring survival. Based on our inferred pedigrees, we found that from all the parent offspring that produce the parent pairs that produce offspring that return and were sampled at the hatchery was 70.7% that were unrelated, while only 14.3% of these parent pairs were between full siblings. We also used the offspring numbers to assess the impact of inbreeding on fitness of parent pairs in different relationship categories. We found the mean number of offspring that returned per mating increased as parental relatedness decreased. We can see here a mean of 0.36 offspring for full siblings, 0.83 for half siblings, 0.88 for first cousins, 1.15 for half first cousins, and 1.5 offspring for unrelated parent pairs. This is very interesting. So what did we learn with this effort? With the age distribution, we saw that more males than females return at age 2, and females commonly return at age 3, and that 2-year-old fish spawn later in the season. This large proportion of 2-year-old fish is important because other studies in wild steelhead show that the proportion of 2-year-olds breeders is small, and this must be considered for hatchery practices. We also saw that total length is strongly associated with age. And this association allows us to suggest to hatchery staff to select larger breeders, especially at the end of the breeding season. Also, we demonstrated that there is a strong genetic component determining the spawning time within the season and also age at maturity. And this high heritability suggests that the age structure we previously observed in many 2-year-old fish could be the result of artificial selection in the hatchery. So, although we see an effect of the level of parent relatedness in the number of returning offspring, our analysis revealed also that only a small fraction of parent pairs, only 3.3%, were close relatives, including the full siblings, half siblings, first cousins, or half first cousins. And since 2013, we proved that the great potential of SNPs for parentage analysis to perform parentage-based tagging to infer biological traits. And with that, it was possible to evaluate the success of the hatchery breeding program by applying this method to the management plans and to find better strategies for the supplementation program. So these large-scale studies show the power of genetic monitoring approach, which meets the goal of most direct tagging programs, but also extends much further, where we can provide family-level information that it is difficult, if not impossible, to achieve with other methods, particularly in these high fecundity species. So now you may be wondering, what use is that genetic tagging approach to me if what she showed us applies to only a very specific system? Well, now I'm going to tell you about this project that is in progress. I'm going to talk about black abalone restoration in Baja California and how we can apply parentage-based tagging. Also, this project is a collaboration between a lot of people, different academic institutions, and in close relation with fishing cooperatives. Well, you know that abalone are gastropod mollusks of the genus Aliotis, and there are 56 species of Aliotis worldwide. In the Northeast Pacific, we can find 7 species: the pinto, flat, red, white, green, pink, and black abalone. So from all these species, black abalone is the only one that is mostly in the intertidal, and its distribution has been greatly reduced in the past 50 years. And a little bit of history of the abalone fisheries in Mexico. Abalone commercial fishing began around 1860 and became one of the most important activities in Baja California for over 50 years, but black abalone was not the target. Then there are only sporadic records of black abalone catches around 1922. The first abalone fishing cooperative was established in 1940, although commercialization of black abalone in Baja California began until the 1950s and reached a peak during the 1970s. Around the 1980s, high mortalities that affected all species of abalone in farms and fisheries were reported after the temperature rises from the ENSO event from 1983 and 1984. And these mass mortalities were extreme in black abalone due to a combination of factors, including obviously the warming events, but also overexploitation and disease, particularly the bacterial infection called withering syndrome. And after that, black abalone in Mexico practically vanished from areas where they were once abundant and is not longer considered for commercial fishing. Then in 1996, an agreement to stop black abalone catches in Baja California was established. In 2003, a report based on commercial catches showed an overall black abalone population decline of more than 98% of the populations. And in 2005, a survey in Baja California found some places with low densities of juvenile individuals. Then in 2009, black abalone made it into the US Endangered Species Act, but there is no official protection in Mexico. And in recent years, black abalone in Baja are showing signs of a slow rebound along some areas, but only some areas. So the goal of this project was to explore the potential of a translocation strategy to recover black abalone in historically abundant reefs. Also, we want to do a genetic monitoring of the black abalone that are being translocated. So 2 black abalone translocations have taken place. Both of them have been led by the fishers from the fishing cooperative. So we're just practically recording what they are doing. All translocated individuals were collected at this place, which is Faro San Jose, and taken to Bahía El Rosario. The first translocation happened in 2021. A total of 125 individuals were collected. 63 were released in Isla San Jerónimo, and the other 62 in Punta Baja. And then in 2023, a total of 600 black abalones were translocated. 300 were put in Isla San Jerónimo, and the other 300 in Punta Baja. How does the translocation happen? Well, first we found the collection spot. Faro San Jose is a location with one of the highest densities that we observed in the late surveys. And second, we had to find the black abalone during low tides. They normally hide under big boulders and crevices. Third, we picked up the abalones using a tool called levantador that the fishermen use. It's like a long metal spatula. And this has to be performed with care to prevent injuries. And during this process, we put all the abalone in mesh bags. Next, we place the collected abalones in empty coolers, and they were transported by land all the way to Punta Baja, where they were oxygenated, and then they were taken by boat to Isla San Jerónimo. And there they were divided into cages. And these cages were put in a mariculture system, suspended in mid-water. And the abalones were kept in these cages for 3 weeks, and they were fed with fresh kelp that was collected in the surrounding areas once a week. And we observed that they started feeding after 1 week of being in these cages, which is a good sign that they're doing good. And we had a 100% survival rate after these 3 weeks, which is amazing. Oh, I forgot that picture. So I would like to point out that these 2 sites had black abalone pre-1990s massive mortalities, but they completely disappeared. We did multiple searches, and none of the sites had black abalones when the translocation happened. So part of our characterization of the site, we did transects with 5 meter long that were 15 meters long, and then we put quadrats every 2 meters. This was part to look for if there were any abalone left, and also to characterize the substrate and to look at the percent cover of algae and colonial inverts, and also to look at all the diversity that was in these areas that we were going to release the abalone. And this is what we find with the site characterization. 76% in Isla San Jerónimo was covered with algae, 11% it was bare rock, and 12% with colonial inverts. Then in Punta Baja, we found 7 species of algae, that's 54.8%. Then 17.8% of bare rock, and 27.4% of colonial inverts. And so before release, we processed all the individuals. We recorded total length and weight. We also sampled all the individuals with this semi-invasive, non-lethal method, consisting on introducing a swab between the shell and the mantle near the anus. So this was to collect mucus and feces for detecting also the bacteria that is responsible for the withering syndrome, and also for future genetic analysis. Finally, we tagged all abalone with physical tags, but they were all lost. So we did also a visual examination to look for symptoms of the withering syndrome, and we didn't find any visual symptoms in the abalone that we were translocating. We also did the genetic detection of the bacteria, and we found that 55.3% were positive for the bacteria. So they have the bacteria, but they are not showing the sickness. They are not getting sick, so they are only carrying the bacteria. And something very interesting is that we found 66.7% of the abalone were positive for the phage, this virus that infects the bacteria that infects the abalone. And we believe that maybe the phage is acting as a natural control of the bacteria in this abalone. Also, these abalone are the ones that survive the massive mortalities, so some genetic aspect of resistance should be acting there too. So then we released all the abalone in these two reefs, in Isla San Jerónimo and in Punta Baja. We normally take the cages there, we put them in the rocks, and we wait for the tide to come up, and then they crawl down and hide in the crevices. So we did some monitoring after these translocations. So the first translocation happened in 2021, and then 10 months after that, we did the first monitoring in 2022. We found 21 out of 63 abalone, that's 33% in Isla San Jerónimo, and 19 out of 62 in Punta Baja, that's 31%, which is also a good number. We probably are underestimating them because it's not easy to find. They are not easy to find. And then in 2023 happened the second translocation, and we went back 13 months after that, and we found 40% of the individuals in Isla San Jerónimo and 19.3% in Punta Baja. These numbers are great for a translocation experiment. Other studies that have done translocations with abalone show about 20 and 30% recovery, so we are in that range. So as I told you, this project is in progress, so we are still trying to monitor and learn more. But what is it about the genetic tagging that I was going to talk about? Well, first we did a genetic analysis in the source population when we collected the abalone that were translocated, and we characterized it genetically, and we found that it's a healthy population genetically. They have a high genetic diversity, which is good for a population that is just recovering. And also we found that acclimation is a fundamental step for the survival of the individuals after they are translocated. But now we want to follow up this population. We think that it's a natural experiment that we can use to learn more about life history traits in black abalones. So first, with the genetic tagging, we want to follow up the population. And also, we are not going to know if this translocation experiment is successful until we get recruits. And if we do the genetic tagging, we are going to be able to know if these recruits come from the individuals that we transplanted, or they're coming from some other population nearby. And also, well, we are going to have more information about the history traits in black abalone. So to consider, so I've talked about molecular methods, and they constitute a very valuable tool for the study and conservation of the species with these complex life histories. But like any method, molecular tools have their limitations, and it is very important to define your goal to establish the correct methodology. I hear a lot of people always telling me, like, oh, can you do the genetics of this? Yeah, what is the goal? What do you want to learn with those genetics? So it's important to define the goal first. Fortunately, over the years, and particularly in recent years, new technologies allow us to have greater precision and increasingly lower costs, so this is great. And despite these advances, there are many situations in which the goal can only be reached if we use other methods. So therefore, the combination of methodologies allows us to understand the general picture for better decision making. And with that, I want to thank all the people that has been involved in these two projects. I will feel like slides and slides of people. It's just a few of them here, and also the funding sources that. Thank you. It was long, sorry. curious on how does that work?
Throw it. And that's the hard throw. That one talk about help and the up like about the black that
Uh,
they're off.
Um, yes, it will take more years. We have an advantage of being able to translocate large individuals because they have a higher survival. I think you can, but it's more challenging to work with smaller individuals for sure, have a higher rate of mortality. So I don't know if it's worth it if we have already these large adults.
Super. I also have a question on the black abalone system. The sites where you've translocated them to, and when you go back to collect and decollect some that are not the ones you've translocated, are you also looking at their genetic diversity and getting an idea of the ones that were originally there? Do they also have a diverse population, and will you be able to then get an idea of, in subsequent years, which ones have been translocated versus which ones might change from the original local population?
So you mean in the local population, if we sample other individuals that were not the ones that we translocated? Yes, we did, and that's how we characterize the genetic diversity of that population. And for that, we use like regular markers. I think we did satellites and SNPs too. But now for the follow-up, for the parentage-based tagging, because black abalone have a very high genetic diversity, 95 SNPs won't work. So we are going to use these other markers that are called microhaplotypes. That is, it's also with the SNPs, but it's a short region, like 100 base pairs that have 4 to 6 SNPs that are going to not go through recombination, so they are inherited directly. And you have more variations in one microhaplotype than just using single. We're going to develop a panel of microhaplotypes to do the parentage analysis in the future. And that's what is going to be used for the pedigree reconstruction. So it's two different things, the genetic analysis for genetic diversity than the parentage analysis. All the way to the back. Throw it.
I have to ask a question.
Oh,
throw it.
there you go.
Okay. So super cool talk. But I was wondering with the stamp, if you have such nice mics, and are you or anyone else at temporal for some of those life history traits that might be assigned? Did you hear my question?
I think I did. I don't think, well, we haven't looked at fluctuations because this is super applied to the hatchery program. We've done some changes throughout the years. For example, in the first study, we found that they spawn a lot of 2-year-olds, so they started spawning a lot. You see the graph of males. Now we have more 3-year-olds being spawned than 2-year-olds. In the past, it was more 2-year-olds. So that changed. Also, they were doing 3 males for one female. That was the spawning hatching. The way they were doing it. and now they are doing one-on-one. And also, recently they started to include wild fish into the hatchery, so not just the hatchery fish that were returned. So those changes have been happening since we started finding these super interesting results in the life history traits. So that's going to change for sure some of the things. No?
Thank you. Great talk. I was curious about whether you had resolution on like what's happening with black abalone at other sites across the range, like probably north of Baja also, because I don't really know what's been going on in like population trajectory of black abalone up here, but I'm wondering if this is some sort of like range interaction with climate.
Yeah, well, they were affected the same everywhere, no? And actually, the reduction the reduction started being reported in the Channel Islands, is where they started to seeing these mass mortalities. So it happened everywhere. And the recovery, I don't know the numbers here in California. In Mexico, we've been doing these surveys, and they're recovering, but very slowly and only in very localized areas. So I think it's going to be the same. And well, here are protected. Down in Mexico, they are not. So sometimes we see a population that is recovering, and this is very sad. And right away, they are gone because they're fished.
Thank you.
Tell me if you have any more. Or you can do it here. Like this.
Oh, okay, yes. Okay. I was wondering, so weird to talk to you so loudly. For the like coupled like spawning time and age of maturity, do you think that they are like related, like just because selection works on those traits like together, or do you think it's like they're like caused by the same molecular like pathways, or like I don't, just always curious, like how does stuff like that happen?
That's a very good.
Yes, sorry.
Yeah, that's a good question. I'm not sure if I know the answer. I think it's a little bit of everything. Like they're being selected together for some reason. They are, how's it called when they come back to the same place that they were